eosinophils, basophils, and all that. And then once this is done, what you can do is find out if T and B cells and their fractions that are present basically, and then this transformed cells, whether it is capable of cell division, it can be seen, whether there are any mitogens that are present. All of this can be seen in this particular leukocyte cell suspension. So when you're taking it, what happens is when you're culturing this peripheral blood, you have to just see that it is obtained in a non-invasive manner, not very expensive to culture. It requires only short duration, and it should be able to yield high quality metaphase chromosomes, and you can be able to reculture from the original sample in up to 4 to 6 days continuously, and this can be cultured successfully 48 hours post-mortem, and it can be mailed as well. So this can be checked for different kinds of issues that crop up basically. So most commonly used mitogen in this kind of cytogenic studies is the phytohemagglutinin, that is PHA. So when this T lymphocytes are exposed to this plant antigen, this phytohemagglutinin is an extract from this red kidney beans basically. So they would respond in the body as a non-cell foreign substance. So mature T cells differentiate into T lymphoblastic cells. Immature ones have the capacity to synthesize the DNA. They undergo mitosis, and after 24 hours of exposure to this phytohemagglutinin, T cells undergo transformation, and as the culture duration increases, it can undergo several rounds of cell division up to 72 hours. And then you have to maintain sterile conditions. Always, whenever you're doing a blood culture or whatever for that matter, sterile conditions maintaining is very important. And also, you have to add an anticoagulant because blood can clot. You have clotting factors. So you have to add an anticoagulant such as a sodium heparin for that matter. For chromosomal analysis, it is one of the best choices. And other anticoagulants interfere with the cell culture. So you've got to check and add the appropriate anticoagulants. And then blood can be cultured up to 4 days after which it is collected. And the temperature, again, it has to be maintained at an ideal temperature. So at around 4 degrees Celsius. And the most commonly used media for blood culturing is Eagle's MEM. Then that is Eagle's minimal essential medium, then MEM alpha, DME, ME medium, then RPMI 1640. Different kinds of media have been mentioned. Ideal pH should be maintained again, that is around 6.6 to 7.8 and between 7.2 to 7.4. And then again, when it is if you've got to add serum again, you've got to add. So a complete medium, again, you have media which is complete and incomplete. So in a complete medium, you have serum also which is added. This can be either from the cow, that is the bovine, or from a horse, or from human serum can also be added. So fetal calf serum and human serum are most commonly used for the cytogenetic cultures. And shelf life is again around 4 weeks at 4 degrees Celsius. And if you keep it at minus 20 degrees Celsius, it is up to 1 year. It is minus 20. In your book, it is given as 20 degrees Celsius. It is not possible. It is minus 20 degrees Celsius. Please correct it in your textbook. And you have to add antibiotics such as penicillin or streptomycin or gentamicin so that, the serum does not get contaminated. There is no growth. And when it comes to harvesting of these cells, you have to see that there are 3 constant. You have to see that, they kind of, you can reproduce the same cells again. So this can be done with following a few protocols. First is mitotic arrest. You use colcimid. Then you can do a hypotonic treatment, that is use low salt concentrations. That is done with solutions of potassium chloride or sodium citrate or salt, different combinations of this. And then you fix, that is 3 parts of methanol to 1 part of acetic acid. This maintains that particular condition. And then you do centrifugation. So centrifugation is done so that you will get different kinds of cell products that are present. So you have a centrifugal force. Basically, they have given that you have the speed that has to be controlled depending upon the product that you're looking at. So you have that, it is known as an RPM, that is revolutions per minute. And you have an RCM, that is the RCF, that is the relative centrifugal force. So RCF, for calculating the relative centrifugal force that has to be used, it is equal to, you have the relative centrifugal force. R is equal to the rotating radius in the centimeters, that is distance from the center of the rotation to the bucket center. So a centrifuge has a bucket in the middle which can rotate, can either go it moves in different directions basically. And then from the center, so you have different kinds of centrifuges. I do not know if whether you will be able to read about it or not. You'll learn it later. Anyway, so that is important. The distance from the center of rotation to the center of the bucket. And then N is the rotating speed, that is the RPM. So for basic serum and plasma separation, you use around 3000 RPM. Basic, I'm just kind of telling you, for example, that is 3000 rotations plus, centrifugation speed that you use. So around 2200 to 2000 RPM for 10 minutes you do. So this is the basic separation speed for serum and plasma separation basically. So once this is done, you start harvesting, and then you have to do mitotic arrest. So that what is done is the first step is to see that the cells are arrested in the mitotic phase, that is the metaphase basically, so as to prevent the spindle fiber formation. So that what will happen is the sister chromatids will be pulled to the opposite side, so that they can be incorporated into the daughter cells. So calcimid, if at all you're adding to that particular cell medium or culture, what happens is it arrests the mitosis, that is production of the daughter cells. So this can be used for harvesting. So you need medium length to very long chromosomes to increase the chance of detecting a small rearrangement, because of which you can tweak the calcimid conditions basically. Also, if at all when you use the calcimid, what happens is you can vary the dosage and exposure time so as to maximize the quality and the quantity of the mitotic cells that are present in that particular harvest. And then so that you can see that again depends upon the culture harvest type and the desired study and the variables that are present. So the condensation effects which occur with longer exposure times can be seen in certain cell types, such as blood, bone marrow, then CVS. All of this have exposure for long times basically. Again, you can tweak the dose of the calcimid to get a increased dosage of the calcimid, so that you can see the mitotic index which increases, straightening of the chromosomes, then chromatid edges. All of this can be seen basically. So some labs, what they do is they prefer the colchicine, but it is slightly more toxic in nature. So what happens is the cell cycle speed is affected. So you will get longer chromosomes. Because of this, it is again a slowing effect. We do not want it. Velban is one more, that is the vinblastine sulfate. It is used for some tissues or all tissues in certain cytogenic laboratories. Good substitute in tumor cultures and difficult specimens wherein, calcimid is not exactly effective in nature. So again, they have kind of given you the chemical additives so as to produce elongated chromosomes. They have given you the different tissue types. Basically, it's lymphocytes, human fibroblasts, and the pleural efficient lymphocytes that are present. The chemicals that are used over here, they have given basically actinomycin D and, different kinds of combination of the aminopterin or thymidine for that matter, or wherein, your ethidium bromide is used. The concentration, The exposure time, is it 1 hour or more than 1 hour, or if it is up to 2 hours or 4 hours is the maximum wherein your mitotic arrest can take place, and the mitotic arrest at what time it is taking place. All of this is given so as to produce elongated chromosomes for much more studying. And then you can do hypotreatment, that is using hypotonic saline solution, so that what happens is your cell volume increases. When you're adding, what do you call it, a hypotonic solution, that is less salt concentration is varying, the cell will absorb more amount of water, because of which the cell volume will be increased, so that you can see the chromosomes spread out in the cell. This hypotonic solution works by creating a concentrated gradient across the cell membrane, and water rushes in because of active transport. And then if the potassium pump is poisoned, that is, if at all the potassium pump that is present on the cell membrane, it is not working properly, what happens is cells don't swell. So you can tweak this also, because of which you can find out the chromatids, whether they are wider or longer chromatids. In different kinds, again, they have given the kind of solution. You can see in page number 200, the formula and what kind of cells can be harvested, whether it is all cells or neoplastic cells, or whether they are amniocytes or fibroblasts or prophase blood cultures, all of this can be harvested. When it comes to the fixation of the chromosomes, what you have to do is, the chromosomes have to be fixed. This process removes the water from the cells, kills the cells, preserves them, hardens the membrane and chromatin, so that they can be banded and fixed onto an cell. So what happens is, you can see the banding patterns also. If if you use the formalin for fixing the chromosomes, you cannot see the banding pattern. But if you use a 3:1 methanol acetic acid fixer, so what happens is, this changes the chromatin structure much lesser than that of the formalin, because of which you can see the chromosome bands. So this is the fixative, which is much easily or commonly used. So this fixative is added slowly, so that metaphase cells don't get lost or broken down. Then once this cell is once the first fixative is added, cells are hardened. Then subsequent fixations can be done, because of which after some time slowly you can actually see the bands that are present. Right? Then you have polypropylene tubes, which resist the fixation action well, basically, so that you have to add additional or perform additional fixation tests for that matter. You will get an RBC contaminated pellets to get rid of the debris. Then if it is present, they will interfere with the slide making and banding. So again, you have to do additional fixation steps. Once after all of this, you have the in situ harvesting. This method was described by Cox and other scientists, wherein, what happens is, the harvested cells are taken, and then they are, the cover slips are harvested in the dishes, removed, attached to the slides for microscopic purposes. So suspension harvests are there, and then the culture is grown. Then dilution with different kinds of RBC is taken and done. Then, you can actually see the amniocytes which are in contact with the culture ware. Then clonal analysis can be done for, finding for aberrations. All of that can be performed. Clonal analysis can be performed. Then morphology of the parent colony that is available. Comparison with the parent colony you have to be done, so that, correlation with the karyotyping can be done basically. Then you will have to take it and then, fix it on the slides. So this process is automated right now, and they're usually inside the culture, wherein, with removable lids. Then analysis of the culture failure is done basically, so that, in case there is any kind of contamination or inability to support growth. Contamination is not the only problem. If at all growth media does not contain enough nutrients also, the culture will not be able to maintain. So you can differentiate it if whether it is because of improperly cultured specimen or inadequate growth or nutrients that are given, and you can follow the preventive measures. So you have this table over here, page number 202. All the cultures are given, the contamination or incubator failure, wrong reagent or improperly prepared reagents, whether there is any kind of cross contamination, and is it because of bacteria, fungi, loss of carbon dioxide, or any kind of mislabeling or anything for that matter. How are you going to do that? So how are you going to prevent this? So you can test the sterility of the media or the sera, then incubator and monitoring all the culture conditions. Then carbon dioxide should be supplied, and then you should see that human and non-human cells are not cultured in the same incubator. So aseptic conditions should be maintained. Then if it is individual cultures, they have given basically the different kinds of causes that can occur, whether it is contamination or incorrect setup or addition of wrong solution or incorrect amount or exposure basically, and what exactly is the result of it, whether it is heat, freezing, or whether there is bacteria produced. All of that is given, and the kind of preventing measures. So to summarize this particular part, you can get to see that media and growth requirements have this physical parameters, that is ideal temperature and conditions to be maintained, amount of gas that has to be maintained, whether it has to be in light or not without any light, and medium requirements, that is you are supplying all the nutrients that is required, all the ions, elements, sugars, amino acids, vitamins, choline, then serum, antibiotics, all of this. And then you're going to maintain aseptic conditions, that is sterile conditions using laminar airflow or hood. Then you're going to use liquid nitrogen to preserve the cultured tissue cells basically, and then you're going to use glass as the original substrate. And the most basic media is the balanced salt solution as given in the example. And you get to see that cell lines required different kinds of serum. So they can be from fetal calf or horse, or for that matter, even from human fetuses. And then contamination, which is actually very rare. And contamination by microorganisms is a major problem basically. So you have to maintain aseptic conditions. Then the cultures can be open or closed type. Then you use peripheral blood basically for determination of the constitutional karyotyping. So the first topic that I had taught you about karyotyping is wherein you use a peripheral blood smear. And then mitotic arrest is, phytohemagglutinin or poplid antigen is the mitogen that is used for helping the cells to divide faster. So it's a cell stimulant in the cultures. Then you have mitotic arrest, and then hypotonic treatment and fixation, which is a necessity in the harvesting procedure. Then you have to use the fixatives, that is the methanol acetic acid in the ratio 3:1. And then you have even automated harvesting that can be done. So this is all about cytogenetic culture and media preparation, and how to maintain this particular setup basically. Now when it comes to slide preparation and staining, I just want to finish this topic also. Maximum possible I will try to finish. Otherwise, whatever is there, I will try to continue in the next class. So more or less, I will just try to share this particular screen. So they've just given basic, as to how to prepare a permanent slide. I'll try to share the screen again. Not much is given basically on slide preparation, staining, and all that basically. So you'll just have to follow whatever is given in your textbook, and I'll try to explain generally. So slide preparation is done for finding out the kind of cells, number of cells that are present, and then to prepare a chromosome slide preparation, you need different kinds of methods that are involved basically. So in this particular topic, you're going to see the process of how to make a slide, the different kinds of steps involved, what are the knowledge, what are the factors that affect the slide making, how is it that you're going to do a good quality slide, and then how to detect human chromosomal abnormalities based on the slides that are there. So again, a brief history of slide making has been given basically. So in this, if you see, you need ideal conditions, which is around 44 to 55% of humidity, and you have to have around 70 to 75 degrees Fahrenheit, which is around, when you convert it, comes to around 24-25 degrees Celsius, if I'm not wrong. One of you can actually see and put in as a message how much is 70 to 75 degrees Celsius when converted into Fahrenheit, 70 to 75 Fahrenheit when converted into Celsius. Then again, these cells are fixed in the 3:1 methanol is to acetic acid ratio, fixative, dropped onto the glass slides, dried, so that you can see optimal chromosomal spreading and morphology. When it comes to the history of the slide, just please give me 1 minute. So when it comes to the history, they have given different kinds, when it started, when all of this started, how they have started putting in the chromosomes between the slide and the cover slip. This was done with a stain which is known as acetourcil, which was a temporary wet mount that was prepared. And alternatively, what happened was they squashed the cells between 50 to 60% of ethanol. And then, this method again, and then this was stuck onto the cover glass. So early slide drying methods involved manipulations such as flaming the cell preparations. So the idea was that it would effectively boil the cell open to spread the chromosomes. Again, you have different kinds of, air drying methods instead of the flaming. Basically, slides were made by dropping the fixed cells onto the wet slides, allowed to dry by evaporation. So today, now you have different kinds of methods for that matter. 2 steps were there. So that is, the first is wherein, they dried the cells by evaporation, and then they found that the chromosomes were spread out in a single plane without any kind of mechanical force that was involved. Now, you have another a person, Nowell, who described the use of PHA for lymphocyte culture, still use the squash method for slide preparation, that is squashing of the cells between the slides and the wet mount. Then it was Moorhead and other scientists who actually modified these techniques to give the slide making that we are using today. Again, fixative is the 3:1:1. Methanol, acetic acid, and chloroform also added, which is known as the Carnoy's fixative. So, so that's what. So you need different kinds of conditions again. Early slide methods involved this flaming the cell preparations and addition of the different kinds of ethanol, acetic acid, and even chloroform. They have just given a lot of that in the history basically. Now, when it comes to slide making, now you have, when it comes to the theory, that is, you have air drying of cells with 3:1 methanol is to acetic acid. And then what happens is, this is based on the theory that chromosomes that are there in the cells, they are enlarged, they have thinner cell membranes before harvest. They will be supported by the layer of the fixative on the slide for the first few seconds after application on the slide. Then after the fixative evaporates, the layer of the fixative becomes thinner. Meniscus pushes down on the top of the cell, enlarging the area, pressing the metaphase chromosomes between the upper and the lower membrane, spreading them out. So this is how you're going to prepare a chromosome slide. Now, when it comes to the protocol of the slide making, Again, different kinds have been mentioned. Please give me one moment and let me see whether I have any other information that can be shared. Um, I'm trying to share one particular, what do you call it, PowerPoint. I'm not really sure whether it is relevant to whatever is going on today, but then at least, we will just get to uh, see basic staining techniques have been mentioned over here, but goals for the slide maker, you are like for metaphase of chromosome, that the fixation and the slide cleaning pertaining to chromosomes has not been seen anywhere. So that is why I'm trying to see whether we have the same anywhere, which is certain times, getting the relevant information is very difficult. Unfortunately, no, I don't think we have this particular. Anyway, you can just have a look in your textbook. And I will have to take like I told you, I just want to finish, or at least finish half of this lesson. So please bear with me till 4:45. I will be taking the class. Unfortunately, no, it is not there. So you will have to look into your textbooks. I think that is the best option that can be done right now. So I will stop sharing my screen. Please look into your textbooks. So when it comes to the principle of slide making, basically what can be done is slide making can be done using a fixative at 4 degrees Celsius overnight. So standard lab procedure, that is glass slides are washed, rinsed with triple distilled water, then kept in ice cold water before the chromosome spreads. Then using a glass pipette, few drops of cell suspension are dropped onto the wet ice cold slide from 30 to 40 centimeter distance. Excessive liquid is drained on from the paper towel and it is kept in a hot plate to dry. So that is one thing. Tubes are sitting overnight. Mix centrifuge the tubes for 10 minutes at around 1200 RPM. Add the fixator, which is around 3:1, 5 ml. Then slides are cleaned, chilled overnight. Then the pellet, which is taken, you have to centrifuge the aspirate and centrifuge the pellet that is present. Resuspend the pellet in the suspension supernated, which is slightly turbid. Then swirl in water, and then you take remove and shake off as much as water that is possible, and then spray, leaving only a very thin film of water on the frosted side. Then once you have done this, you have to place around 2 to 3 drops of the cell suspension across the horizontal, that is across the slide. Then place it on a water bath. Then blot the edges of that particular slide basically using a paper towel. Finish drying them, and then what you have to do is you have to adjust or finish drying according to the relative humidity that is necessary. Then whatever painted edges is there, you have to label. Labeling is very important when you're making a slide for that matter. Lab number, date, slide number, so that, and you have to indent it also so that you will know what kind of slide you have prepared. Then look at the slides under a phase contrast microscope so that you will get to see the mitotic index, chromosome contraction, chromosome spreading that is necessary or that is required. So then with the phase contrast microscope only you can see all of this. Then when finished making this, you have to collect the pooled cell pellet in small tubes, add around 2 to 3 ml of fixator for long-term storage. Again, different kinds of technique variations I've mentioned, wherein you can differentiate or increase the humidity or decrease it, and uh, basically try drying the slide using the hand or the warming plate for that matter. Then you can decrease the humidity, place it on a stack of wet soaked paper towels, completely making it dry, and other conditions basically. Then you can check in the phase contrast microscope. Now, when it comes to the goals of the slide maker, you're going to see that mostly gray to dark gray chromosomes are seen. Light chromosomes yield trypsin-sensitive cells with poor contrast between the bands. Very black images may have increased the cytoplasmic background and they are resistant to trypsin. Light chromosomes scatter so that you can see basically broken metaphase conditions everywhere. So this causes mosaicism. This is seen in mosaicism, or it can actually mask the true mosaicism, which is again low, very low percentage is seen. So these are the different kinds of issues that can be identified with the slide. Minimal number of chromosome overlaps for accurate counting and band analysis is done. Very thin cytoplasmic background is, if possible, so that trypsin G banding will be optimal. So that also should be taken place. So they have given a figure over here in your textbook wherein metaphase spread as a function of the duration of the drying time you can see basically. So that's given. So A, B, C, and D are there basically. So you can actually see that picture. I'm trying to share one picture right now with you, if possible. Just a second. Share screen. Share. Right. So this is how under a slide you can see different kinds of metaphase chromosomes basically. So some you can do a chromosome banding as such. So basic drying on the slide, without any kind of mechanical force will actually align the chromosomes in this linear manner. So metaphases that dry too fast are always tight with many overlapping chromosomes. That dry at optimal rate do not have any few overlaps and they're not broken. That dry too slowly are characterized by both metaphases, and by tight rolled metaphases you're going to get such kind of a picture basically. I'm hoping. And this is a G-banded metaphase that dried too fast. It's encapsulated in the cytoplasmic membrane as such. So what happened was chromosomes were not yet spread out completely. Trypsin is uneven due to the interference by the proteins. So this is the kind of pictures that you get basically. Whenever you're doing different depending upon the kind of drying fast or drying slow. And when it comes to the importance of each factor in slide making, you have to see the slide type. The cleaning of slides is also important, and how you do centrifugation, what kind of fixative you're using. So just by changing the type or the brand of the slide, what happens is, you can see that, whatever chromosome picture that you're getting can change. So you've got to clean the slide also properly. Any oil on the slide will actually ruin the spreading of the chromosomes. So you have to use a good quality slide marker to label the slide. That is also important. Centrifugation. at ideal conditions, that is 2 ml microcentrifuge tube, round bottom, centrifuge at around 6000 rpm for 2 to 5 minutes in a tabletop microcentrifuge, and 14,000 rpm for around 2 to 2 seconds. Further helps pelleting all the cellular material so that you get only the nuclear material which is necessary for you. These high-speed centrifugation tubes should not damage the cells or the chromosomes. So you have to maintain the chromosomal material for both G binding and the fish protocols. So when you're working with these small precious pellets, you have to be careful because you're going to get it in the pellet form. This pellet is again taken, harvested, cultured. You follow again the hypotonic treatment in the same tube, addition of fixative, and then you're going to add it onto the slide. Fixative is again the methanol and acetic acid, that solution is added, and then methanol actually dehydrates the cell, acetic acid removes the histone protein and the nucleic acid. So only the non-histone protein will remain. This is essential to the binding of the potential chromosomes. So again, common fixative is, that is used in the cells, so that, the cells are kept in the swollen form. So it is a hypotonic solution that is necessary. And then also the number of fixatives, the fixative ratio, quality and the freshness of the fixative also affects your metaphase chromosomes. Again, it is given in your textbook basically. So the number of fixatives is how many times you're using the fixative. And multiple fixation can actually strengthen the cell membranes, improve the chromosomes morphology, get rid of the debris such as the red blood cells, lysed red blood cells basically. And then when it comes to the fixative ratio, that 3:1 ratio has to be maintained basically so that it yields optimum chromosomal banding quality, preserves the cell properly. And then again, for G banding contrast, you can use that is what you can use G banding contrast to fixative also for the G banding purposes. And when it is high acetic acid, that is 2:1 or 1:1 methanol is to acetic acid concentrations, higher acid concentration improves the spreading because it increases the fragility of the cell membrane, decreases the speed of the fixative evaporation. So you've just got to check this before fixing it onto the the slide basically. You can use ethanol to acetic acid fixative also instead of methanol. So again, different kinds of based on the toxicity, and you can use ethanol which is much less toxic than that of the methanol. So then the quality and the freshness of the fixative is important. And mixture also, what happens is it is hygroscopic in nature, so it absorbs water upon standing. So if you use freshly prepared fixators, the more better slide stain preparation you will have. Angle is also important while you're dropping the the cells onto the slide. So at a 20-degree angle from the top, if you actually add from the cells are placed in the upper third of the slide. I don't exactly have a picture. Yes. And then you add it like this at a 20 to 30-degree angle, and then you just inject it onto the slide. This should allow the downward movement so that, what happens is cells don't pull up in one side. And then you have to see that, you just kind of center it. So effect of the dropping of cells on the slides along this log acid is axis is because water in the fixative drain from this may cause faster evaporation, because of which that should be maintained. Greater tilt angle helps in speeding up the drying process. Uniform drying can be achieved by tilting the slides at one angle from the part of the drying time and another for the final drying. Then you have to see the dilution factor also, which is necessary. Because if you dilute it more, then you will get very sparsely spaced cells. If you dilute it less, all the cells are in only one place altogether, crowded cells. They do not spread or they do not band properly. And you have to do controlled chromosome spreading also. Standard protocol again, smears that is taken out, and then you have to see for how it is being spread, all the chromosomes. And then you have to check for consistency of the results. Are you getting the same picture after you have stained again? So that should be checked using culture media. Again, you can use GCT2 culture and then the EB culture for that matter, using this, because you're going to maintain that particular culture as such. And then you can see whether you're staining and seeing if it is there's any kind of differences in the way the metaphase chromosomes are spreading. And again, the cell and the culture type differ. The cell type is that you use all certain cell types. Basically, they slide better onto the the glass slide. Basically, all the uncultured direct harvest, such as the bone marrow, CVS, solid tumors, they have or they're more difficult to spread onto the slide. Blood specimens are the easiest to make a slide in between, and bone marrows tend to be the more difficult ones, with amniotic fluid specimens, which are slightly in between. So again, harvesting techniques and the cell drying techniques are important over here, wherein the cell culture variables, that as micro versus macro blood cultures, the number of bone marrow cells used for inoculum, primary versus secondary, subcultured, harvesting techniques that is the type, amount, duration, temperature, all of this make an impact on the culture. When it comes to drying, again, you have to see whether after before you drop, what is the temperature that is there. Basically, what is the slide temperature, this also impacts. And whether the slides are water soaked, so that variable humidity, and this impacts the drying process as such. Slides can be soaked in the water prior to pipetting the cell suspension. Then you have to see that if there is too much water on the slides also, this prevents the chromosomes from spreading while drying. Then fixative on the slide is also we have already seen acetic acid and how it is to be dropped. You have got pictures of the same, in fact, in your textbook. You can just have a look at it. Different pictures from 8.6 onwards, how the fixative starts drying. Of course, very bad pictures, but unfortunately, these are the ones. If at all I find any kind of pictures available or any kind of PowerPoint regarding the same, I will try to share it with it. Right now, I don't exactly have the same. So you can just have a look at your textbook. How the mitotic cells lose their halo, starts flattening, because of which chromosomes spread faster than the non-mitotic cells. Chromosomes become dark and visible. And then the next one is when the halo of the light is still there. Nuclear membrane or content is more resistant to the flattening forces of the fixative. Then on the fifth point is that tiny puddles of fixative are surrounded that are present. They quickly evaporate. Visible chromosome spreading stops. Minor changes are also seen. And then based on these experiments, you can see, the height of the cell suspension has no influence on the chromosome spreading. It is just the angle, it is just the amount of fixative, amount of diluent, that is the diluted factor. All of these make an impact on the amount of, how the chromosome is being fixed. So aging of the slide, again, if you see 8.8 to 227 page number, you get to see that, it is subjected to dry heat or ethanol. So slides are allowed to sit for around 3 to 4 days, and then they are suitable for the banding purposes. So they can be G-banded, that is gene sustaining, with the different kinds of karyotyping basically. So the aging process is very important, so that you can find out the G-banding or the Q-banded chromosomes basically. So the major change in the chromosomes from the aging is because of the oxidation of the protein, which is sulfahydryl groups basically. Protein sulfahydryl groups which are there, they get oxidized, because of which chromosome banding can differ. Then again, this aging will actually cause the degradation of the chromosomal DNA. So this is the reason why some banding methods and fish methodologies are affected by artificial aging or by slide heating. And then you can store it in around 20% of AEC in the jars with around 100% of ethanol. It will stay for around 1 to 2 days in the ethanol and does not require any kind of prior aging before you do the fluorescent in situ hybridization techniques basically. And slides solely for used for G banding can be stored at the room temperature. but you can also use 18 to 24 month slides for fish analysis. And you can also use 6-year-old slides for that have been stored in ethanol for fish results. Then washing again has been mentioned over here with adding water and T-Pol, which is a washing detergent, and then soaking the slide for around 20 minutes. Then replacing with normal water, washing again, cleaning the surface, transferring the clean slides, storing the beakers in the refrigerator for overnight, and these slides can be used for slide making. So slide making and the staining methods have been given over here again. We've already finished seeing the Giemsa staining and the different kinds of staining methods. Do you want me to tell you again the different kinds of protocol? Or else I will just kind of share it with you in the next class if it's possible. So I will stop the class for today basically. We've already seen the staining methods, that is the chromosomal banding methods. That is the Q banding, the R banding, the T banding, the G banding, and the C banding. So I've already shared those particular slides with you. Videos being
yes.
So I will not be covering it again. So you can actually just go through. In case you have any kind of doubts regarding the same, you can always ask me. But these have been explained to you in the last class, in the first few classes, in fact. How these, and the preparation for G banding in this technique, I will just explain it to you in the next class if possible. Right now, I will just stop the class. And in the next class, we'll just go through the microscopy, the different kinds of imaging, what exactly are the parts of the microscope, what exactly is lens magnification, all of these. And then maybe, go for and the glossary part, that is the genetic terms and all, you can just have a look at it, just for your perusal. They have given appendix A and B, different kinds of malformations. So in the next class, what I will do is I will talk about microscopy and imaging and developmental biology. I will start. All right.
Okay, madam.
Thank you so much.
Thank you, madam.